Bismillahirrahmanirrahim. My name is Dr. Imran Wahid. Today I will be starting a new lecture on renal physiology, that is the physiology of kidney and urine formation. But before I start, I request you to please subscribe this channel and click the bell icon so that you can get the notifications about my videos and lectures. In this lecture, I will discuss the fundamentals of renal physiology, and in this we will discuss about the basic gross and histological anatomy of the kidney, and we will discuss about the nephron, which is the functional unit of the kidney, and we'll also discuss about the glomerulus, the tubules and the peritubular capillary networks, and all the histological and gross anatomical features of the kidney. So let's start with this. Kidneys are known as chemists of the body, and they are two bean-shaped structures, and they are located on either side of the vertebral column in human beings. Kidneys are very important for regulation of the electrolyte and acid-base balance in the body, and also for excretion of the waste products in the urine. They are also responsible for maintaining the overall hemostasis and chemistry of the body. Now, so far as gross anatomy of the kidney is concerned, we can see that if we section the kidney longitudinally, a section of the kidney along its long axis, when it is sectioned, we can see that it is, as I said, a bean seed shaped structure. You can see this. This is the outer portion. This is known as cortex, renal cortex. And the inner, this is the dark portion. The inner one is renal medulla. This is the light portion. So here is the renal pelvis and ureter. This is the ureter. This is renal pelvis. And here, these are the dotted small granules, and this portion is dotted and is coarse because of the presence of glomeruli in the kidney cortex. This portion is known as medulla, where we can see medullary pyramids. And at the tips of these pyramids, we have calyces, major calyces and minor calyces, and the tips of the papilla, they open into the minor calyces, and they then open into the major calyces and into the urinary pelvis, and it ultimately drains into the ureter. This is the gross and these are the gross features of the kidney. So as I mentioned, you can see there is a darkish reddish brown portion on the outer side and or the peripheral side, and this is known as cortex. And a deeper, lighter portion, this one is known as medulla. Medulla is again divided into a number of conical or cone-shaped areas known as renal pyramids. ये renal pyramids हैं. Renal pyramids. They have their broad base toward the cortex, and their apex is pointed towards the lumen. This apex has small sieves project into the minor calyces, which we will discuss later on. The lateral boundaries of these are formed by renal columns of Bernardi, name of the scientist. The apex of the papilla, or these minor calyces, they have small perforations in them, and these perforations are known as area cribrosa, which is perforated by a number of collecting ducts or tubules, which open into the minor calyces, and then they open into the major calyces, which ultimately open into the renal pelvis. Nephrons are the functional units of the kidney. A nephron is the functional unit of the kidney. Every kidney has about 1 million nephrons in the human body. There are two types of nephrons basically. superficial or cortical nephrons, and these superficial or cortical nephrons which are present in the cortical region, they occupy outer 2/3 of the cortex. Superficial nephrons, they constitute about 85% of all nephrons in the kidney. As I mentioned, nephron is the basic functional unit of the kidney. Second category of nephrons is known as deep or juxtamedullary nephrons. They occupy the inner 1/3 of the cortex and constitute about 15% of the total nephrons in the kidney. Under normal physiological conditions, the superficial or cortical nephrons or glomeruli are fully functional and they are smaller in size, whereas these deep or juxtamedullary nephrons, they are large and work only in conditions of stress. So now let us discuss about the nephron. What is nephron? As I mentioned, nephron is the functional unit of every kidney. Kidneys are made of functional units, and just like neurological system, we have neurons. In kidneys, the functional units are nephrons. There are many functional units in every kidney, and the functional unit of the kidney is known as nephron. There are about 1 million nephrons in each kidney, and each nephron is made up of two parts. Nephron made up of two parts. One is the Bowman's capsule, and second one is the renal tubule. Bowman's capsule basically is a dilated blind beginning of every nephron which surrounds a tuft or bunch of capillaries known as glomerulus. Bowman's capsule and glomerulus, they together constitute what is known as Malpighian or renal corpuscle or Malpighian corpuscle. It is the name of the scientist who discovered it, the Marcello Malpighi. Bowman's capsule has a specific function that it is involved in filtration of all the non-colloidal constituents of the plasma, while the tubules, renal tubules, they are responsible for reabsorption from this filtrate. All the essential substances are reabsorbed and sent back to the bloodstream. The non-essential things are not absorbed and they pass out in the form of urine. So these are important facts you need to remember. The tubules, renal tubules are very important. They are involved in the formation of urine, filtration, secretion. There are different types of tubules, the proximal convoluted tubules, the distal convoluted tubules, and collecting ducts. They join up through a system of connecting tubules, and they together join to form minor calyces which project into the apex and pill of the renal pyramids. Minor calyces join to form major calyces which ultimately open into the renal pelvis. So if we follow the flow of the filtrate fluid from the glomerulus, once the fluid is filtered from the glomerulus into the urinary space, it enters into the proximal convoluted tubule, then loop of Henle, then it passes to the distal convoluted tubule, then into the collecting ducts, minor calyces, major calyces, and finally into the renal pelvis and ureter, and the urine flows out. So this is the flow of the filtered fluid from the glomerulus. The fluid is filtered in the glomerulus, proximal tubule received it, entered into the loop of Henle, distal convoluted tubule, then collecting ducts, they join to enter into the minor calyces, they enter into the major calyces, and they flow the fluid into the renal pelvis and the ureter. Now let us see what nephron actually is. What is it made up of and what are its basic structures? So let us see in this diagram very clearly that basically this is a diagram we need to understand. You can see in this picture very clearly that this is an artery which is entering into the glomerulus. This is known as afferent artery, afferent arteriole, and this is coming, the blood is coming out to this, and this is known as efferent arteriole. This is known as a Bowman's capsule. It is a cup-like structure. You can see cup-like structure, artery is entering into it, and this is divided into multiple capillaries, and at the efferent end the capillaries come out. Blood enters into the glomerulus from this site, circulates in the glomerulus, and comes out through the efferent arteriole. This is the distal convoluted tubule. This is the Bowman's capsule. It has two layers, a parietal layer. This is a parietal layer and a visceral layer. The parietal layer is lining the outer side of the Bowman's capsule, while the visceral layer is in contact with the bunch of capillaries endothelium, that is the glomerulus. So it contains, basically Bowman's capsule contains the glomerulus and the Bowman's capsule. Bowman's capsule and glomerulus together they form of the renal corpuscle or the Malpighian corpuscle. So as we see, this is a cup-like structure which surrounds the glomerulus and collects the filtrate fluid and small molecules that is filtered and forced out of the blood by pressure of the circulating blood in the glomerulus. Both the glomerulus and this Bowman's capsule, they constitute together as Malpighian or renal corpuscle, as I already discussed you. So this blood filters out of the capillary loops through the glomerular basement membrane. This is the glomerular basement membrane and podocytes and into the urinary space. The space between the parietal and visceral layer is known as urinary space. So once again concentrate on this diagram, and you can see that this is in efferent arteriole. Blood enters into the glomerulus, circulates, come out to the efferent arteriole. This is distal convoluted tubule which is in contact with the efferent arteriole. This is the Bowman's capsule, outer parietal layer, inner visceral layer in contact with the glomerulus, which has podocytes and basement membrane of the glomerulus. So this is together the Bowman's capsule and glomerulus is known as renal corpuscle or Malpighian corpuscle. So this is the picture of the Malpighian corpuscle or renal corpuscle. It is very important to know that this Malpighian corpuscle is actually the first step in urine formation, filtering waste products and excess substances from the blood. Blood filters out of the capillary loops through the glomerular basement membrane and podocytes and into the urinary space. Again, a glomerulus is important, so we need to further discuss it. There, the glomerulus and Bowman's capsule, that is the this Malpighian corpuscle or renal corpuscle is the essential component of the nephron, which is functional unit of the kidney. Glomerulus is the network of capillaries and it has endothelium in it. It acts as a filtration unit, while Bowman's capsule is a double-walled structure which surrounds the glomerulus, as I have shown you, and collects the filtered fluid. Initial filtration of the blood takes place in the glomerulus. So this is the first step in urine formation. It is basically specialized, but glomerulus is basically a specialized capillary bed where blood enters through the afferent arteriole and after circulating exits through the efferent arteriole. The afferent arteriole is in contact with the distal convoluted tubule. We will discuss this later on. What does it mean? The high blood pressure within the capillaries of the glomerulus forces the fluid and small molecules like water, salts, glucose and waste products out of the blood into the Bowman capsule's urinary space. So what is filtration barrier? I would like to introduce another word, filtration barrier. As I told you, this is the glomerular basement membrane which contains the podocytes and the endothelium of the glomerular capillaries and epithelium of the visceral layer of the Bowman's capsule. So endothelial cells, mesangial cells and podocytes and specialized epithelial cells, they constitute what is known as filtration barrier. Filtration barrier means that it is a barrier which does not allow the unnecessary materials to pass out through the kidney. So it is important to understand that filtration barrier is responsible for all these features. Blood filters through the capillary loops through the glomerular basement membrane and podocytes into the urinary space. So again and again we have to concentrate on this picture. Again, it is very important to know that kidney has, as we have seen that kidney has functional units, nephrons. So until and unless we are aware with the nephrons, this basic glomerulus Bowman's capsule, we are unable to understand the base of the glomerulus and capillaries. So let us discuss further and move on with the physiology of the nephron, other portion of the nephron, that is the proximal convoluted tubule and other tubules. But before that, let us see another diagram to clarify the renal corpuscle further. This is a better diagram, much better explanation of renal corpuscle, and here you can see very clearly that this is a renal corpuscle. Here he has shown us that this is the afferent arteriole, this is the efferent arteriole. Blood enters through this site and comes out from here after circulating in the glomerulus. These are glomerular capillaries are shown, and the visceral layer of the Bowman capsule is in contact with this along with podocytes and their foot processes. The foot processes of the podocytes and especially the epithelial cell, they constitute the glomerular basement membrane, and this distal convoluted tubule comes in contact with the afferent arteriole. The cells of the afferent arterioles, it undergoes modification to form JD cells and cells of this distal convoluted tubule that constitute the cell known as macula densa. Both the JD cells and macula densa constitute what is known as juxtaglomerular apparatus. We will discuss it later on. Currently, we can see that it is the Bowman's capsule with its parietal layer, visceral layer, glomerular basement membrane with mesangial cells and podocytes. And this is between the parietal and visceral. This is the urinary space where the filtered fluid enters and goes into the proximal convoluted tubule. So now let us see the structure of the nephron. This is the structure of the nephron. Nephron is the functional unit of the kidney. You can see again, this is the afferent arteriole, this is the efferent arteriole. Blood enters from here into the renal glomerulus, circulates and is exits through this efferent arteriole. This is the glomerulus and Bowman's capsule, what is known as renal corpuscle. From here the filtered blood enters into the proximal convoluted tubules. It is known as convoluted because it has folds or convolutions. From here the filtered blood moves down into the proximal convoluted tubule, and from here into the descending limb of the loop of Henle. This is the descending thick limb, this is the descending thin limb, and then it continues into the ascending thick limb of the loop of Henle until it reaches the distal convoluted tubules. Distal convoluted tubule is also has convolutions, that is why this is known as convoluted tubule. From here it enters into the collecting ducts. To further clarify, we can see that collecting ducts are shown here, and from here we can see that another diagram, we can see that this is the description of the previous diagram in a better way. Efferent arteriole, afferent arteriole, and this is the interlobular artery. Lobules are present between the renal pyramid, and this is the interlobular artery and interlobular vein. From these arteries arises a network of capillaries which surrounds the glomerulus and the renal tubules. You can see that is the network of capillaries. This is known as peritubular capillary network. The purpose of this network surrounding the glomerulus and the tubules is that the distribution of the fluid electrolytes between the fluid flowing into the nephron, various portions of the nephron, proximal convoluted tubule, and descending limb of the loop of Henle, ascending limb of the loop of Henle. The exchange between the blood and the fluid in the nephron takes place through these capillaries. So this is the peritubular capillary network. Afferent and efferent arterioles reliefs and afferent arteriole enters the glomerulus. Blood is filtered and is poured into the fluid is poured into the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and collecting ducts. And this peritubular capillary network throughout continues around the tubule of the kidney to facilitate reabsorption and secretion of the materials. The filtered fluid then moves down into the collecting ducts. Collecting duct moves on the tip of the collecting duct. The collecting ducts open into the minor calyces at the tip of the renal papilla, which open into the major calyces, and they ultimately open to the what is known as renal pelvis. So here we can understand that renal fluid, the fluid which is filtered by the glomerulus, it enters into the kidney glomerulus, it circulates into the glomerulus, and from the glomerulus the filtered fluid enters into this urinary space, and then entered into the proximal convoluted tubule, loop of Henle, distal convoluted tubule, and from here into the collecting ducts, and there into the minor calyces, major calyces, and renal pelvis, and flows out as urine. Bowman's capsule is very important. Visceral and parietal layers have special structure. Basically the parietal layer is simple squamous epithelium, if we speak histologically, outer wall of the capsule. Visceral layer is made up of podocytes with foot processes, pedicles, and they interdigitate to create filtration. The space between the parietal and visceral layer is known as urinary space. Just to revise it moves the fluid into proximal convoluted tubule. The filtration barrier is formed by the glomerulus. The Bowman's capsule is crucial for maintaining proper kidney function and removal of the waste from the body. So let us revise a few words about the renal cortex and renal medulla. As I mentioned, this is the gross anatomy. So before we leave the topic, it is important to know a few points. The cortex is the outer layer, as I mentioned, and medulla is the inner layer. Both play an important role in filtration and regulation of the blood. The cortex is the outer portion of the kidney. It contains glomeruli, proximal and distal convoluted tubules, and peritubular capillaries. That is why this is granular and darker. It has a grainy appearance because of the presence of glomeruli and convoluted tubules. The initial filtration process takes here, and the filtered fluid entered into the urinary space. So the structure of the kidney is very important to understand gross structure. Why this is why this is granular on the outer side, and why this is smooth on the inner side. The reason is that cortex contains, you can see, it contains these grainy structures which are due to the presence of glomeruli and tubules in the iris structure. Therefore, it appears darker and granular because of the presence of glomeruli, convoluted tubules, and peritubular capillaries. Whereas medulla, it is the inner portion of the kidney beneath the cortex. It contains loop of Henle, collecting ducts, and vada recta, the straight arteries, specialized capillaries. It is smoother than the cortex due to collecting ducts opening into the renal calyces. The medulla regulates the urine concentration and plays a vital role in water and salt balance, whereas initial filtration takes place in the cortical region. So this is very important to understand what is actually meant by this cortex and medulla. So before we leave the topic, let me discuss a few words about what is known as juxtaglomerular apparatus. As I told you, that juxtaglomerular apparatus is a specialized portion of the kidney, and it plays a vital role in the kidney renal physiology, regulation of blood pressure, and regulation of all these features of the regulation of blood pressure and salt, sodium retention, sodium and potassium excretion, etc., and various matters related to the electrolytes. So juxtaglomerular apparatus, you can see again in this diagram. This is very important. As I mentioned to you previously, that when the cells of the efferent arterioles, they enter into the glomerulus, Here is the site where they come in contact with what is known as distal convoluted tubules. The cells of the afferent arteriole which are in contact with the distal convoluted tubule, they undergo some changes and they are converted into granular cells or JG cells, juxtaglomerular cells. They produce a substance which is known as renin, whereas the cells of the distal convoluted tubule, they are converted into cells known as macula densa, and macula densa are very sensitive to sodium concentration. So juxtaglomerular apparatus is a specialized structure in the kidney, crucial for regulating blood pressure and glomerular filtration rate or GFR. The afferent arteriole coming in contact with the distal convoluted tubules, both these elements, that the afferent arteriole cells and distal convoluted tubule cells, undergo changes, and distal convoluted tubule cells are converted to macula densa and arterial cells are converted into what is known as JG cells. Macula densa are specialized cells in the distal convoluted tubule. They sense the sodium chloride concentration in the tubular fluid and they play a vital role in regulation GFR by communicating with juxtaglomerular cells. So juxtaglomerular cells are present here. They are very important. They are present in the afferent arteriole wall and synthesize what is known as renin. It is an enzyme that initiates the renin-angiotensin aldosterone system. We can see another thing, that is the extra mesangial glomerular cells. These cells are located in the angle between afferent and efferent arteriole. In this angle we find extramesangial cells, extraglomerular mesangial cells between afferent arteriole and macula densa, between the afferent and efferent arterioles and between the macula densa. The angle between afferent and efferent arterioles and macula densa, in this space we find extraglomerular mesangial cells. They are important, very important role in communication and coordination with JGA cells. Renin-angiotensin aldosterone system is the juxtaglomerular apparatus is central to this system and it helps to regulate the blood pressure and fluid balance. When sodium concentration is low, for example in hypovolemia, macula densa signals JG cell to relieve renin. Now what is the tubuloglomerular feedback? This mechanism involves the macula densa sensing changes in sodium chloride concentration and adjusting glomerular filtration rate accordingly, helping to maintain GFR. You can see that JG cells, they produce renin. You can see in this diagram, another beautiful diagram, renin-angiotensin aldosterone system. Renin is produced by the JG cells, and you see in this that this renin, it acts on it is an enzyme which acts on a protein angiotensinogen which is produced in the liver and converts it into angiotensin 1. Angiotensin 1 is again acted upon by angiotensin converting enzyme and converts it into angiotensin 2. Angiotensin 2 is then degraded by the angiotensin enzyme and released into circulation. Angiotensin 2 has important effect. It stimulates the adrenergic system, causes sympathetic activity, when tubular sodium chloride reabsorption and potassium creation and retention. So when there is a hypovolemia, macula densa assesses the low concentration of sodium, gives signal to the JD cells, they produce renin, and angiotensin 2 is produced by this mechanism. Angiotensinogen is cleaved into angiotensin 1 by the renin, and angiotensin converting enzyme converts into angiotensin 2 which is produced in the lung, angiotensin converting enzyme. So we can see here that sodium, potassium, chloride is sodium is reabsorbed, potassium is lost. So there is activation of the aldosterone system which causes sodium retention and potassium excretion. Adrenal gland produces aldosterone. So this is the effect of aldosterone. Angiotensin is a potent vasoconstrictor and causes arterial vasoconstriction. Similarly, pituitary gland releases ADH, antidiuretic hormone. So there is reabsorption of water from the collecting ducts. It means that sodium is also retained in hypovolemia and low sodium concentration. Sodium is retained, reabsorbed, water is also reabsorbed, and there is hypovolemia, which was hypervolemia, to correct the hypovolemia which was caused by low concentration of the sodium. So this is the tubuloglomerular feedback of the renin-angiotensin system. So with this I conclude the lecture, and in the next lecture I will discuss the physiology of urine formation, what is the composition of blood, what changes take place in the glomerulus, how it is filtered, and how many changes that flowing fluid from the proximal to distal contour tubule, loopal, fenlec, collecting ducts take place, which converts this filtered blood into a fluid and which flows out as urine. Once again, watch the video till end, subscribe the channel, and click the bell icon so that you can get notifications about the videos. Thank you very much.